The lac operon in bacteria is kind of our standard example of how an operon works. Lac stands for lactose, which is one of the sugars that E. coli can break down. Now, before we get too far into it, I want to say that an E. coli cell is going to prefer glucose. So if there is glucose present, the E. coli cell is going to preferentially use glucose. So we are talking about an operon that encodes genes to break down a secondary type of sugar in their environment, lactose. Now, in general, the genes for breaking down lactose are going to be turned off because if there's no lactose present in the environment, there's no reason to have the genes hanging around that are going to be able to break down that lactose. So, as long as there's glucose, we're just going to use glucose and the cell is happy. But sometimes there is not glucose present. And so in order for the cell to keep going, it has to metabolize something else. E. coli can metabolize lactose because lactose is a disaccharide made up of glucose, which is a monosaccharide, and a second monosaccharide called galactose. Galactose is a similar sugar to glucose, but it's not quite the same. It's a little tiny bit different. So if we have lactose present, the first thing we have to do is break down lactose because eventually we want to get to that glucose because that's our preference here. And with any disaccharide sugar in any organism, they're going to have to be broken down first into its smaller components. So if there is lactose present but no glucose, then the cell is going to let lactose in, and it comes in by a little channel protein called a permease. This allows that lactose to come in, and then we start our process of breaking down this sugar. The first thing that happens is an enzyme called beta-galactosidase. We're going to call it BGAL for short. Beta-galactosidase is an enzyme because we've got that ase at the end. We know it's doing something in the cell. Converts lactose into a quick intermediate. And it can go either way. It can just directly break it down into galactose or into glucose, but it can also break it down into this little intermediate. And there's a reason that it needs to be broken down into that little intermediate first. And we'll talk about that. Once we get some of this allolactose, then we can just convert the rest via beta-galactosidase into galactose and glucose. So we can do either way. We need a little bit of that allolactose to get started. So the lac operon is going to be our operon with a promoter at the beginning, followed by an operator, and then 3 structural genes. So we won't worry about those just quite yet. The operator overlaps the promoter and the 5' end of the first structural gene. So what it's really doing is we've got our promoter sequence here at the beginning, and then we've got this sort of gray area operator sequence or region that overlaps with our first gene. So remember that the operator is the site of binding of our regulatory protein or regulatory gene. So it's going to bind at that operator. So if there's no lactose present, the cell is happily chugging along using its glucose. The lac operon has a repressor bound to the operator. So the lac operon is a negative inducible operon. This means that we have that regulatory element bound to the operator to block transcription, which means that it is inducible. It is by default off, but needs to be turned on when lactose is present. So let's talk about how this happens. All right, so we need a couple of things. First is we need an inducer. An inducer is going to cause the release of the regulatory protein from the operator. So we need the inducer first. So the inducer is that molecule that I mentioned, allolactose. Remember, when lactose comes into the cell, it meets beta-galactosidase, or BGAL, that begins the breakdown of lactose into glucose and galactose. But it can first produce this allolactose, is sort of an intermediate molecule. And when there's any allolactose, it's going to act as an intermediate. Once we get some allolactose and it begins its job as an inducer, then beta-galactosidase can convert can convert lactose into galactose and glucose. So there are a couple of genes that are going to be needed for this. One is the LAC-I gene. This is the repressor. Remember that a repressor binds that the operator region and blocks transcription. And remember, this is normally off, so when there's no lactose present, our genes are not expressed. We are also going to need the Operon promoter and the Operon operator genes as well. All right, so then we need our structural genes. Remember, our structural genes are the active genes that are working to express whatever protein that we need. So once there is some lactose present, we need to produce more of that LAC-Z gene, which is the gene for beta-galactosidase. Now remember, beta-galactosidase's job is to convert lactose, first to allolactose and then to glucose and galactose whenever it's ready. So truthfully, in the cell, there's always a little tiny bit of beta-galactosidase floating around, just a little bit, because if lactose happens to enter the cell, beta-galactosidase can go, oh, look, there's lactose. Let's turn this on. So we need just a little bit always, but in general, it's going to be essentially off. We also need the LAC-Y gene. Now, the purpose of the LAC-Y gene is to produce the permease, which was that channel that lets lactose in. So a little bit of lactose gets in the cell, and then suddenly the cell says, woohoo, we need to convert all this lactose. We need to let more in, and we need to convert it. And to do that, we need permease to let it in and beta-galactosidase to break it down. And then there is a LAC-A gene. LAC-A is, for our purposes, function unknown. Actually, in general, the function is unknown. There's some theories on what it has to do. We're just going to say that it is there. It probably has to do with some sort of detox of the cell. that we are not going to worry about. So there's also this LAC-A gene that is going to get induced as well, because remember, this is an operon and we're expressing lots of genes all at once. As I mentioned a minute ago, that LAC operon never quite fully shuts down, just in case a little bit of lactose is ever present in the cell. So here we've got our whole LAC operon. We have got that promoter region and the regulator region on our gene, followed by our set of structural genes. Now remember, the lactose operon is a negative inducible. Negative meaning that transcription is blocked by our regulator protein binding to that operator site, and inducible meaning that it is normally off and needs to be turned on. Remember, it is on just a little bitty bit, but we're going to generally say that it is turned off. So when there's no lactose present, that repressor is bound. We don't need much of that beta-galactosidase or the LAC-Y gene. We're not worrying about the LAC-A for now, and so this is generally shut off. But once a little tiny bit of lactose comes into the cell, beta-galactosidase is going to convert it into allolactose. Remember, allolactose is our inducer, and that inducer molecule is going to bind to our repressor protein. So that little repressor that is keeping this gene from being bound. So once we get a little bit of lactose, that means we get a little bit of allolactose, which means it can now bind to our regulator protein and force that protein to fall off. Once it falls off, once it is no longer bound to that operator, then RNA polymerase can come in, express beta-galactosidase and permease, which we need because remember that beta-galactosidase is going to break down lactose into galactose and glucose. So we're going to start producing more of it, and once we turn off all of these regulators, we don't really need any more allolactose. So beta-galactose is just going to convert directly to glucose and galactose. And then we need that permease, because if the cell realizes that there is lactose, then it's going to need to bring more in, because if it is decided to have a preference for lactose, it probably means there's no glucose, and so the cell needs to get glucose by breaking down lactose. And so it's going to start making more permease, which is going to act as that little channel to bring in lactose. So once a little bit of lactose is present, we essentially knock off that regulator and then allow transcription to continue. So here's a bigger picture of our LAC operon. It's the same one as on the other slide. I would suggest reading through carefully, taking some notes on what's happening to all of these molecules. It is relatively confusing, so I would suggest that you use some of the extra additional videos that are posted in Brightspace to help you a little bit. But as always, I am more than happy to chat with you before or after class or during office hours. We are going to use this for our CRISPR lab. We use a modified lactose operon as a screening tool to see if our bacteria in our CRISPR lab have taken up the gene that we want it to. And we do something called blue-white screening, not just for CRISPR, but for other molecular projects and biotech projects. So it's a common tool for studying bacteria, and we're going to utilize this LAC operon for that.